Record. We're recording. Terrific. Hey everybody, Dr. Rob Silverman here, and I am with Chief Science Officer of KBMO Diagnostics, Dr. Brent Dorval. How are you, Brent?
I'm doing well today. Thank you very much, and thank you for everybody on the other end of the line.
I'm happy we could make it here. I'm happy to have you. We're going to talk about the rapid antibody test. So before we do that, why don't you tell everybody a little bit about your background? It's quite extensive.
Sure. Again, my name is Dr. Brent Dorval. I'm the Chief Scientific Officer of KBMO Diagnostics, a Massachusetts company located in Hopedale. And I am one of the group of managers of the company. James White is the owner and president of the company, and Anthony Recupero is the Chief Operations Officer. KBMO Diagnostics was founded in 2004 to capitalize on a patent that I received for measuring food sensitivities / food allergies. Now, with the current problem of the COVID-19 virus, we had to kind of switch gears, and we retooled, and now we're using a rapid test to measure COVID-19 antibodies, IgM and IgG, to this particular virus to aid in diagnosis. My background, I've got about 35 years in diagnostic development. I invented the first HIV rapid test, 1-minute test for HIV. And also, I was a visiting scholar in the Department of Chemistry at MIT for a few years, and we did some work with polio virus. So viruses per se are no real stranger to me. It's just, I guess this is an opportune time to reinvestigate some of these viruses, and COVID-19 is one of them. So the assay that we're offering is the COVID-19 rapid test. And this, as I mentioned, measures IgM for early infection or IgG for late infection. Now, there are 4 main proteins for the COVID-19 virus that people are concerned with. There are many more, but the main one is the spike protein, which the coronavirus gets its name for the spike protein. It's a round virus, about 150 nanometers. These projections from the surface make it look like a corona, hence they call it the coronavirus. So you've got the spike protein, you have the membrane protein, which is a major structural protein, you have an envelope protein, and then on the inside, the subject of this discussion will be the nuclear protein, which forms the nucleocapsid. So you have a helical positive stranded RNA inside the virus that's like a beaded pearl necklace. Along the strand of the mRNA is nuclear protein, which is just decorates the RNA. So the test measures the antibodies against nuclear protein. And you may say, well, why would we choose nuclear protein over some of the other proteins on the virus? The antibodies to nuclear protein come up the earliest and are the strongest. So first, in general, the first response you get is IgM, probably in the 5, 7, 8 day range, followed by IgG against the nuclear protein. And that happens probably around 8 to 15 days and really gets strong out about 2 weeks, roughly. So what happens is the IgG titer far outstrips the IgM. So IgM is produced early. It's in low titer, means there's not much around. It has low affinity, and it's not a very good neutralizing antibody, whereas the IgG antibody is produced a little later. It's produced in much, much higher titer. It's very, very strong binding and provides good neutralization of this virus after you've so-called been infected and gone through that phase. The way the neutralization occurs is mainly through binding to the spike protein. So it blocks receptor binding and prevents entry of the virus into the endothelial cells, alveolar cells, and a variety of other cells as well. The way this test works is very, very simple. It's a card, and it's nitrocellulose membrane, and it's striped with the nuclear protein. And what you do is you apply a sample either 10 microliters of whole blood, serum, or 20 microliters of whole blood to each well. And as the sample crosses the test line, it will bind, the antibodies bind. And then the colloidal gold will bind to those antibodies. And if you get a red line at the test line, then you know you're positive. And the sample continues on and forms another line at the control line. So when you look at this test, you'll either get no lines, the control line means you're negative, or you'll get 2 lines, either at the IgG site or at the IgM line, and that will mean that you're either IgG positive, IgM positive, or both. It's a rapid test. Generally within about 5 minutes, you can clearly see that you're either positive or negative, but we let the test run to 15 or even 20 minutes just to make sure that nothing is showing up at all. The nice thing about this test is it's based on the nuclear protein, and probably people say, okay, there's a whole bunch of different types of coronaviruses, so what's special about this particular? Won't it pick up other viruses maybe that you've been infected with in the past? And the answer to that is no, because this particular antigen, nuclear antigen that we use, is a truncated nuclear protein, and it's specific for SARS-CoV and CoV-2. So we'll only pick up the current pandemic strain of virus, which is very nice. Now, there are other tests out there. They cross-react with a whole bunch of different types of coronaviruses. So it's hard to determine, are we measuring something in the present tense, or are we measuring something that's happened in the past tense? And that's important, because when you look at the IgG response of coronaviruses in general, the IgG response can be detected out there 10 or 12 years. So we know that we had 2 pandemics, one back in 2014, and also 2004, so that they were much smaller, more defined, but nevertheless, there would be a high likelihood that some of the other tests out there would pick up antibody to past strains and really wouldn't be relative to the current pandemic. So that's just an important point. Now, the other test you're probably going to see out there in conjunction with testing clinically is the PCR test, the real-time PCR test. Now, that's a very, very good test because it picks up the viral genome. The problem is people don't like nasopharyngeal swabs. They feel it's a bit invasive, and a finger stick or a blood draw is much more palatable. The issue with the PCR test is they only pick up about 34% of the infections. Now, that may be that they're sampling the wrong site. It may be a temporal thing where they're either too early or too late in infection, and they just don't pick up enough of the virus or any of the virus so that they can do the PCR on it and detect it. So that's an important factor when you think of that. You'll see, especially in the news, 2 prominent tests: the Abbott, which is a PCR test that just came out. It's a 5-minute test, but in conjunction with that, what I'll share with you in the near future today is why it's important to measure antibody along with PCR, and you can up your chances of clinically confirmed cases of really good detection. So if we just take a look at some of the clinical data. Now, this test has been used on 2 million patients in China and at about 20 different hospitals or clinic settings. So this is something that's tried and true. It has been approved by the Chinese CDC/FDA, their regulatory agency, and currently it's been under review at FDA, and we're expecting approval in the United States based on that large cohort of data, but the FDA is overwhelmed, understaffed, and given the current conditions, they may be a little behind the 8 ball, but they're saying 2 to 3 weeks we should have an answer on this. And that's very important because a lot of tests out there, and I'm not going to tar everybody with the same brush, have not been clinically tested, have not been clinically proved, those antibody tests. And as a result, yeah, you take a test, the results are sometimes equivocal, the results are not accurate, and therefore the clinical usefulness of the test is limited because of that. And I've had a lot of experience in rapid testing over the years, and what I can tell you about this assay is when we first looked at this. So we partnered up as a distributor with this Chinese company because as KBMO Diagnostics, we've got enough scientific staff. We could have cloned the protein. We could have made our own test, but we don't have 6 months or a year to make the test and then prove its clinical utility. So we chose the quickest route that would be the most benefit to all the patients and doctors out there would be to partner up with proven technology, a clinically useful test that's been demonstrated to work in a large population. So we took a quick route to market, so to speak. And in this particular test, what they found was when in that huge cohort of patients, there's really kind of 3 groups of people when you look at this. The first group is PCR positive and antibody positive. And when you look at that group of people, PCR picks up about 34% of the people. So, okay, that's not bad. However, when you combine that with antibody testing for IgM and IgG, you pick up another 40%. So when you look at the combined overall sensitivity of this particular test, if you do PCR and antibody, it goes from 34% PCR positive to about 74%, 73% if you do both together. So clearly, you're upping your chances, and it's a much more clinically useful test because those patients that have been infected, you're not picking up with PCR, and if you do the dual measurement, you're picking up those patients combining with the antibody test. Now, it gets even better when you look at the second group that's PCR negative, but they're antibody positive. And of that group, this test picks up about 96% of those. So as a physician, I think that would be extraordinarily important. You're saying, okay, the gold standard, PCR, these people are negative. Wonderful, right? Well, the story gets a little more complicated than that. If you measure antibody, 96% of those people are picked up as antibody positive. So clearly the take-home message there is that when you look at this under with clinically diagnosed patients, that the data you get is much more useful if you combine it with an antibody test. And the last cohort they looked at was there's a minor group, about 25%. And I think if you read in the literature, they're going to call them patients that have fever of unknown origin. So they're patients that have displayed some sort of fever, and fever is certainly one of the hallmarks of this particular infection. Yet it's due to something else. It could be due to a staph infection. It could be due to some other illness that would generate a fever. But it's not due to COVID-19. And in that case, they're both antibody negative and they're PCR negative, but they're clinically probably it's responsible for some other type of illness that's associated with that. So with that, I'd just like to share with you that you can get this particular test at KBMO Diagnostics. The blood draw is available now, and we'll be launching the finger stick, which will be the 903, the Wattman card, where you can just do a finger stick, put a drop of blood on filter paper, and mail that back to the lab. It's cheaper, quicker, and patients really like that. The price of the serum draw is $100 plus $50, and that includes the overnight shipping to the client and back. And the payments are required before we ship the test kit. The nice thing is the turnaround test for the time for this test is 24 to 48 hours. And currently the volume has just been astounding for this particular test. And if you're a provider, then just go ahead and set up an account through KBMO Diagnostics. We'll post all your reports to box.com. It's like a sophisticated file folder. You'll have your own file. You put your password in, and you'll be able to access all your patient reports. and they'll be uploaded again within the 24 to 48 hour time frame. Now, if you're a patient and you need phlebotomy services, we have also arranged with a network of about 6,000 phlebotomists to be available to either you can go to them or they can go to you and draw your serum. I think you'll need the order of one of the healthcare, a nurse practitioner, a doctor, something like that. You can't call up as a patient and get this test ordered, but the doctor certainly, if he or she thinks it's prudent, can get that particular test for you. And just one other thing I will mention is we're going to be coming out with another test very shortly for the spike protein. And it's a unique spike sequence that's on the little stalk that's the binding site for the virus binds to cells via, they call it the ACE2 receptor, the angiotensin converting enzyme receptor on like alveolar cells and other types of cells in the kidney and endothelial cells. So what's important is that if we can measure antibody to that spike protein, what we're thinking is if we do that in conjunction with measuring the N protein, then we'll be able to probably pick up even more individuals itself. So what it will do is give the clinician more information to base a diagnosis and treatment on. And one thing that is coming, that just came out pretty much in the past couple of days that was kind of interesting in that regard is that people in the IgG phase of the illness were thought to be pretty much immune, and that's true to this particular strain. What's coming, what's emerging is that these people still may be shedding some virus. So what's important, even if you're antibody positive, especially IgG positive, is to work with your clinician and you know, everybody, the goal is to get back to work, to see your family members again, and to re-enter society and get back to normal. I think that's what everybody wants to do. So there's a judgment call is what I'm saying between the time you test IgG positive and then when your healthcare provider thinks it's prudent for you to re-enter society, so to speak, and circulate again. And it probably will even depend on the state and the stage of the pandemic in that particular area. So that's what I would like to say with my closing remarks. So thank you very much, everybody. I really appreciated this time with you.
Wow, that was great. That was a ton of great information. So I'm going to splice it out for everybody. So I'm going to take you back to the beginning. You said it was an RNA virus, correct?
Yes.
And it has spikes. They call it corona because it's like a crown and has spikes. But isn't the spikes a little different because aren't they 3? Whereas
Yes.
the previous ones are not?
Yeah, it's well, the spikes are, it's a heterodimeric trimer. So there's a mouthful. So yeah, each spike has a S1 domain and an S2 domain. So you can think of 2 little domains on the outside of the cell. And then that spike is associated in trimers. So there are 3 of those. The outside end of the spike, furthest away from the viral membrane, that's the receptor binding domain that actually contacts the receptor and makes the first contact. So the S1 part of the domain is responsible for the dimer. The other part of that dimer then undergoes rearrangement when it's clipped by the enzyme that's associated with the ACE2 receptor. So it binds, the spike gets clipped, S1 is released, S2 undergoes a rearrangement, inserts itself into the host cell membrane, and then viral fusion and entry of the virus occur right after that. So that's what happens to the spike, is proteolytically degraded by a host cell protease, and that enables it to enter the host cell.
And that protease is furin, which is very unique. That's never happened before. You talked about antibodies. Antibodies, so we have our body, we have an immune system. We actually have 2 facets, as everybody knows. We have an innate and we have an adaptive. So innate's great. It's got these natural killer cells and everything, and they come out right away. and you spoke about two of the antibodies, IgM, which you said was early, and IgG, which was kind of later. And IgG is sort of the big guy because it's 75% of your immunoglobulins. And you had told me when we talked before, and I want you to reiterate it, that IgG actually blocks the ACE2 receptor site. Having said that, so everybody knows ACE2 receptor sites are prominently on the lung, but they're on mucous membranes, so they're also on the gut, the liver, and the kidney. And that's why you see this particular virus being so virulent, because it can attack multiple organs and tissues at the same time. But I'd love you to talk about the antibodies just for a moment and what that means, and how like IgM is good. It's sort of like a pawn, but the king or queen is the IgG because of its properties.
Yes, well, IgG, again, it's a little later in developing during the infection process, but the titer is much, much higher. It's much, much longer lasting, but mainly the affinity, meaning the ability to bind, is much, much higher. So what happens is the virus infects you. You're producing antibodies, early antibodies against the nuclear protein. We know that. We can measure them. Now, a little bit later on, you're also getting antibodies to the spike protein, and it may just be temporarily a couple of days. But what happens is those IgG antibodies against the spike protein bind to the receptor binding domains on the S1 spike. So on the spike protein of the virus, the antibody binds and it sterically inhibits that S1 protein from binding to the ACE2 receptor. So if you block the binding of the virus to the receptor, then you don't get any proteolytic conversion, and you won't get any infection. And then IgG is another interesting thing. If that happens, then it's forming an immune complex. So that's an interesting thing, because then it activates complement, and you get decorated with C3b. And if that happens, then you get the reticular endothelial system. So that's just a fancy way of saying the spleen and some places in the gut. Will start to remove these immune complexes along with the virus and break them down and degrade them. So as a result, the IgG has that secondary, that's a part of the innate immune system that's just there. But once that IgG binds, forms an immune complex, then the body wants to remove that complex and degrade it. And that, in fact, is what happens.
Well, great explanation. We all know that, I mean, Fauci said antibody testing, Gupta said antibody testing. In two sentences, why would we want antibody testing? I agree, I took it. And
yeah.
We'll talk about that.
Well, yeah, it's one more, for a couple of main reasons, really. Number one, if the patient is exposed, especially doctors, especially people on the front lines, in our case, a friend a young lady that was exposed to her father who had COVID, they want to get antibody tested because just a sheer peace of mind. I mean, it's like, oh my God, I know I was in proximity. I'm not feeling terribly bad, but you know, am I safe to move around? Do I have to self-quarantine? What's the situation? So it's one more parameter that antibody testing is one more parameter that a clinician can use to say, hey, look, you're either antibody positive and definitely been exposed, or you've been exposed but you're antibody negative, and we'll test you in 2 more weeks or 3 more weeks just to see if you seroconvert. In other words, if you produce antibody. The second reason is that, as you see with the clinical data, if you PCR test somebody, you're going to pick up 34, 35%, a third of the patients. If you then test with antibody, you're going to pick up another 40%. So that will, in an epidemiological perspective, that really tells you the number of people that have been infected or seroconvert, because PCR doesn't tell the whole story, and neither does antibody. But when taken together, I think they paint a much, much bigger picture. And then the last is that if you do get infected and you seroconvert to IgG, much like with other viruses and much like with vaccines, it's a very good thing. I mean, in many ways. That's an indicate that you're immune. Now, that doesn't mean that you can re-enter society immediately, whether the workforce or walking around. That's a consultation between the patient and the healthcare provider. But it does mean that you're coming into the convalescent phase of this disease. You've produced the IgG antibody, and at least insofar as this strain, you're immune.
So let's go a couple of scenarios so everybody knows. We talked about antibodies. We're testing for IgM and IgG. Let's say patient comes and is IgM positive, but IgG negative. What do you say?
In that case, you're in the early phase of infection, so you've definitely been exposed. You're seroconverting. You're in the early part of the infection. And what I would do at that point is I would say, look, we're going to have to wait another couple of weeks to make sure that at that point, you'll about 90% will seroconvert at about 15, 18 days. So you want to wait it out and get IgG tested. For sure, you'll be positive at that point. And then you'd probably want to get PCR tested again. I know, again, there's some question of whether you should or you shouldn't, but it's certainly a conversation to be had with the healthcare provider as to, look, I've seroconverted, I feel great. Do I have to stay home or what's the situation in that particular state? So that IgM would just indicate an early phase and you're kind of transitioning into IgG.
Okay, second scenario, you're IgM positive and you're IgG positive.
Right, that's the best news a patient could receive, because you're going through the early phase. The antibodies, what they call class switch, from an IgM class to an IgG. That tells you you're transitioning from an early phase to a later phase, and that you've seroconverted to IgG, which indicates that you're starting the production of neutralizing antibodies. You're starting in the phase where you're actually blocking the virus from binding to the receptors and preventing infection. And you're also starting to clear this virus from your body. And that will happen in a later phase.
How about negative IgM, positive IgG?
And again, that's the best scenario for a person that's been infected. You're now in the convalescent phase. You're producing high titer of antibody. The virus is probably effectively being blocked from entering the cells. It's being cleared from the body. And it's going to take a few days for you to get your strength back, or for your lungs, to heal, or bronchitis. So you'll have some sequelae that will occur that may linger for weeks, maybe months, depending on the status of a patient, whether they're immunocompromised, whether they're young, whether they're healthy, that sort of thing. But you'll be getting better, but it may be a longer convalescent phase for certain individuals as compared to others.
Now, so here's what I did. We took the test, we ordered it to speak to what you said. We ordered it one day, got it the next day. Phlebotomist came a couple of days later. The phlebotomist, we don't want anybody in our house. The phlebotomist we let in, she was wearing a mask, wearing gloves, she was wearing a gown, and I happened to have some footsies for her. So she came in, all covered up, came downstairs. And when she went to take the blood, she put on this fiberglass looking mask. So she coughed on us. Now, my wife and I, we were wearing gloves and we wore masks for her protection.
Yes.
We were. So it went really well. We've really been careful. So no problem in letting her come back if I have to take the test again. I took the serum test. Now, I took the serum test on Friday. I got an email that you guys received it on Saturday, next day. I just got my results today. We're filming on a Sunday, and within 24 hours, and both my wife and I are negative IgM, negative IgG, excuse me. Of course, I wanted to have some. So having said that, that would mean that I have no antibodies yet. So I have to, now that reading would say I still have to be careful because I'm still susceptible.
Yes.
Correct?
Yes, that's how I would read that, because you were exposed to somebody that was positive for that particular disease. So just because you're antibody negative today. That's a snapshot in time. It tells you today you're fine or you haven't been infected. But what will happen if you seroconvert days later? So you're right. It will probably be prudent to have a test probably in another 7 to 10 days.
Which we'll do. And I think what we'll do is we'll probably follow up with another test, or we'll follow up with the blood test. Now,
Right.
here's the question. And I don't want to use me just as an example. Could someone have been exposed, literally had the virus, and it didn't do anything, and yet not produce any antibodies? So better, let me rephrase that. Could mine or somebody's defense mechanism be so good that I don't have to produce antibodies, but if you get hit with the virus and it gets in your nasopharynx, you have to produce antibodies?
Yeah, I mean, I think most likely it's the latter. You know, if you get exposed to this virus such that it's really going to set up an infection, you know, it's, in other words, viable virus. I mean, if you can call a virus viable, but I mean, certainly functionally viable virus. I would say that you're probably going to get the virus at some point. Now, if your immune system's really good, maybe you could clear it more quickly. But I would say the likelihood is that you're going to get replication and you're going to seroconvert at some level.
Yeah, we need that. So interestingly enough, you're specific for COVID-19.
Yes.
Where some others, we won't say who, labs may be testing for coronavirus, which is a vast number of viruses, and probably me negative. Probably if you tested for that, it may have come back positive because I probably at some point was exposed to one of those viruses over the last 12 years, whatever it may be.
Could be.
Right. So this is a much more precise test, and with the subjects up to about 2 million kits.
Yes, that's correct.
Correct. So N is 2 million versus others who just started and maybe 100, 500, or 1,000. So that's why you feel so imbued with enthusiasm and scientific certainty that this is a very accurate test.
Yes.
One thing I want you to, because I know you had said with the PCR and you had said with the test, let's talk about something, sensitivity and specificity, and let's just say that one's a positive, a false positive, and false negative.
Okay.
So everybody knows. They don't know those two technical words. Say what they are and talk them about.
Okay.
What your test is.
Yes, a false positive simply means that you're really negative, but the test itself is going to give you a positive result. So instead of getting no line on the test line, you get a faint line or some line shows up. So while that is not a deal killer, you still have to be around high 80s, 90% to have a really good test. In this particular test, the first set was 87% and the second cohort was over 90 for the sensitivity of this particular test. So that's very, very good. And when used in conjunction with PCR, that goes up even higher. Now, the deal killer is if you get false negatives. You never want to get a false negative on it. You can accept a false positive because clinically now you can rule that in or out, but if you get a false negative, you're saying the patient is not infected when in fact they are. And that could have dire consequences, much like the work we did with HIV. The deal killer in a test like that is if you are telling people they're not infected and they really are, they're free to go about their business and they're going to infect other people. And what you want to do from an epidemiological perspective is kind of err on the side of false positive if you have to, and definitely not have any false negatives. That, like I say, that would be a deal killer. And in this particular, the specificity was 100%. So that's a pretty powerful number. And anything in life's hard to be 100%, but in that particular study, it was 100%. Another study that just came out that was a preprint that I saw, it was 99%. So what that and it was with using this test. So what that tells me, and that cohort was 305 patients. So a smaller cohort, but nevertheless very carefully controlled clinical cases. that tells me this test is working well. You don't see you don't see those numbers if a test isn't put together well, if it's not functioning well, meaning you look at it and the reads have to be clean, crisp, and unequivocal. You can't be saying, you're looking at it and you say, hey, Joe, come over here. What is that? Is that a positive or a negative? So you can't tell, but you're looking for somebody else to kind of confirm something. And I see a lot of tests like that. If you have questions like that when you're running this test or those tests, they're just not functioning very well. It has to be clean and clear.
Absolutely. And I really wanted you to get to that because, having discussed this, the fact that it's virtually 100% where you're not going to get a false negative, which would be bad, meaning that patient is positive, they think they're not, and they're going back into society and they are not, which means they're just going to infect everybody. I mean, we just saw yesterday in South Dakota, 190 people, because they didn't know one or two people had it in the confines. And we're not picking on anybody, infected up to 190 people with a smaller state. So that was one of the things that really enticed me to want to go with this test. I'll just make a little statement. It's funny, as a functional medicine chiropractor, you originally had talked about the food inflammation test. And right now, it's not a bad idea to test for food inflammation, because the whole point to keeping yourself healthy and not getting what we've talked about, this cytokine storm, is not to have too much inflammation. So if broccoli, for argument's sake, you were inflamed to, it would elevate your inflammation. You need to take it out. So even though right now everybody wants the antibody test and they should take it, we all know that they're calling it for it on the TV, you can still do some other things as you do your telehealth. I'm excited about that spike protein test. I'm excited about the blood test. So those who take the blood test are those who are afraid of needles, correct?
Yes.
Those who are under 6 or 7, the kiddies.
Younger children or even infants, yes.
And those who just absolutely don't want a phlebotomist to come anywhere near them, they are cocooning appropriately. So, and that test is almost as accurate as a serum test, correct?
Oh, it will be as accurate. We just got some very exciting data out on Friday, and I don't want to get ahead of myself, but what we're doing right now is we're setting up a study where we'd like to test 100 positives and 100 known negatives, randomize them, and test them on blood spots. So we're going to do spike recovery, and what we know is that we can spike negative serum, negative whole blood, and we can recover all the antibodies. So we figured out how to extract it. And that sets the stage for us to do a small clinical trial to prove conclusively that if you send in a dried blood spot to us from a finger stick, that we're going to get the same result as if it were whole blood or as if it were serum or plasma. And the test is so good, we just don't want to put out something just because it's easier and cheaper. We don't want to put out something that functionally isn't as good, isn't up to our standards. So we're taking our time, but we fast tracked everything, and we should have some exciting data coming out probably the mid to end of this coming week.
That's outstanding. So somebody who's got a negative, so you can use my blood.
Great.
And I'm excited about the finger spot. And the reason I took the test is I want to know where I was. And I've recommended, as you know, to all my patients, I said, I think you should take this test. It tells you where you are. So I'm negative, so I still have to be careful and everything like that. I was hoping to have IgG for a couple of reasons. And it's a good thing if you have IgG. If I'd gone through IgG feeling the way I did, it would even be a better thing. What I would do with the IgG was, those are the people who may be able to go back to work sooner and get into society in a confined place. Also, and I know you don't have to speak to this, the IgG would enable me to use some convalescent plasma. I'm not looking to sell it. I'm looking if my mom got it, my wife got it, or somebody I knew that wasn't doing really well with it, that I could give my blood plasma. So it's a really good thing as a starting point. Many of my patients have taken it. I was just a little early. Just got a text. I looked, somebody already told me they got their results. I just didn't want to fiddle with my phone. I think this is a great thing. KBMO Diagnostics. If you're a practitioner, I actually have a code. My code is COVID-19 /DRRob. And any patients who need a physician, just give me a call. I'll get you right in there. Any docs who have any questions, reach out to me. I'll get you right to Dr. Dover or James White, who's the CEO, and you guys can ask me questions. And it's been my pleasure.
Great.
Thank
you very much.
Thanks for taking some time on Easter Sunday. I greatly appreciate it.
Thank you. Have a nice day.
You too now.